Good morning, everybody. So I think maybe we start. My name is Axel Hutt. I'm the chair of this session, and I have the pleasure to introduce Kristin Sellers. Kristin works at the University of California in San Francisco with Edward Chung, and she works in the field of translational medicine. Before, she has been working in the University of North Carolina with Flavio Fröhlich and worked with animals, with ferrets, in the context of neurostimulation and anesthesia. And today, currently, she applies neurostimulation in humans with a focus on major depression and closed-loop neurostimulation. So it's quite exciting, and it's an interesting approach, very close to clinical work. And interestingly, I can mention that her work raised some attention of non-scientific journals. So San Francisco Chronicle and the New York Times reported about her work. And so we are looking forward to your talk.
Axel, thank you very much for that introduction. It's a pleasure to be here. My second time in Australia. Always take an opportunity to come visit this part of the world when I can. Many thanks to the organizing committee. It's really great to be back in person, see people face to face as compared to those little squares on the screen. So today, I'm just going to get started. I have no disclosures relevant to the work that I'll be talking about. So overall, what I work on is neurotechnology development for clinical applications. There's kind of three main areas to my work. The first is novel probe and array development. This is done in collaboration with Lawrence Livermore National Laboratory and IMAC, the manufacturers of neuropixels. So here we work typically in the intraoperative setting, trying to increase the density of the types of recordings that we can do both on the surface of the human brain as well as in deeper brain structures. I also work on a program developing an implantable device for long-term chronic recordings in the ambulatory setting and accompanying clinician software. And then the focus of today is going to be talking about clinical trials, looking at personalized closed-loop deep brain stimulation for the treatment of major depressive disorder and chronic pain. Today we'll be focusing on that major depressive disorder indication. And across all of these programs, there's really a huge interdisciplinary group of individuals who come together from their different backgrounds, different training regimens, but everyone's coming together to focus on the brain. And I really appreciated Joseph yesterday introducing the flow diagram of theory to tools to applications. So today I'm going to be focusing more on the application side of things to hopefully spur thoughts and ideas for how we can take that to feed back to adapt theory and then update the kinds of tools that we have in our arsenal to apply to these questions that we have. In this case, to really inform clinical treatment for individuals who are suffering from neuropsychiatric disorders. So very briefly, our roadmap for this morning is going to be outlining the unmet clinical need that we're currently trying to address, going over a bit of background on deep brain stimulation for major depressive disorder, at which point I'll introduce the Presidio clinical trial, which is the clinical trial that we have for treatment-resistant major depressive disorder. And then in the context of that trial, I'm going to use some of those data and things that we've learned from those patients to walk through 3 vignettes of key challenges that we're facing and how to progress individualized psychiatric treatment. So we're going to talk about where we are, how's it going, and is it working now? And then lastly, I'll have a few brief closing thoughts about looking ahead. And for each of these vignettes, I'm going to be raising some questions after I go through the data that we have. Just take this as a starting point for brainstorming. This is really supposed to be, an information transfer session from the clinicians, the experimentalists, to people who think more about the computational tools and analyses that we have. So it's really just supposed to, spur conversation as compared to the very specific questions that I bring up. I'm the one who has the privilege to be here to talk about this with you today, but there's a huge team back in San Francisco and across the world with our collaborators. I'd really like to highlight how well this team works together, and I'll be highlighting the individuals who are key players for the different parts of the story as we go along. So overall, there's a huge unmet need for the treatment of neuropsychiatric diseases. So depression is one of the leading causes of disability worldwide, and nearly 300 million people in the world are afflicted. The standard of care currently includes pharmacotherapies of a number of different varieties, psychotherapy, the combination of both of those, and then additional treatments such as electroconvulsive therapy or transcranial magnetic stimulation. However, despite these different options, approximately 30% of patients fail to respond to these standard interventions. So this opens the door for additional therapies, and one of those is deep brain stimulation. So when we think about deep brain stimulation for major depressive disorder, there's a number of questions that first come to mind. First, where should we stimulate? The neural circuitry underlying major depression is not fully understood. We know that it's a network disorder. It includes both cortical and subcortical targets, but in terms of getting on the highway that this network is part of, we don't know if there's multiple on-ramps or if we really have to be in a specific point. And furthermore, the etiology of depression in individuals is highly varied with different constellations of symptoms. So the same diagnosis may present very differently across individuals. And that leads us to think that the best targets may actually be individualized. So thinking about personalized medicine and personalized therapy. Currently implemented deep brain stimulation strategies involve continuous stimulation, but many people who have depression actually experience varying symptoms. So we have a mismatch between the timescale at which we're applying the therapy and the timescale at which these symptoms are occurring. Do we want to bring those timescales into harmony? Also, with continuous stimulation, individuals are getting stimulation during sleep. How is this disrupting their normal sleep? Is it disrupting their normal sleep? Is it beneficial for the overall therapy of depression? These are questions that we don't have answers to at this point. Also, with continuous stimulation, we think about neural adaptation. This is the same stimulus applied continuously, and we know the brain basically responds to novel stimuli, so there's a large chance for adaptation. So taken together, these lead us to think that closed loop or responsive therapy may increase efficacy of this treatment, lower the risk for side effects, and then in more practical terms, reduce need for battery replacements and other considerations of the like for actual clinical application. And lastly, there's also questions about dosing. So most MDD pharmaceutical treatments take about 4 to 8 weeks to become effective. And it's a bit unclear why that's the case. The half-lives of these medications don't line up to the 4 to 8 week timescale. So there's an open question about how we should be dosing electrical stimulation in this context. So deep brain stimulation overall, I'll say here, being a US institution, we often think about the FDA in terms of approvals, so I do apologize that this slide is going to be very US approval focused. In 1997, the FDA approved deep brain stimulation for the treatment of essential tremor. In 2002, treatment approval was granted for Parkinson's disease. And then following this, in 2005, there was a first seminal study in the field by Helen Mayberg's group that showed remission in depression of 4 out of 6 formerly treatment-resistant patients. And this was achieved by stimulating in Brodmann Area 25, the cingulate. And I want to highlight that the patients who are eligible for trials such as these have failed pretty much literally every known treatment to date. So these are the sickest of the sick. These are not people, kind of your median patient who has major depressive disorder. So following these very promising results, the Broaden trial started, which was a multi-center, randomized, double-blind trial for deep brain stimulation. There was huge excitement at the onset of this, but unfortunately, a few years later, the trial was terminated as part of the 6-month futility analysis. And during this interim analysis, they showed a 17% remission. So while technically the level of remission did not require termination of the trial, the funders and other involved entities decided to stop the trial at that point. However, a number of the patients continued to receive therapy. They had implanted devices with batteries that were still viable, and there was a 24-month follow-up, at which point 50% of the active stimulation patients dropped their depression scores by at least 40%. Again, these are people who had no relief from any other prior treatments. So taken together, this indicates that there's still promise for deep brain stimulation in the context of MDD, but there's something that we're missing. Was it specifics about the cohort that was recruited for these trials? Was it where the stimulation was being applied, the regime, the duty cycle of the stimulation? Something that we need to figure out the answer to continue the utility of approach like this. So over the years, there's been a growing number of different targets for stimulation in terms of where we should stimulate. So Helen Mayberg's group first talked about the subgenual cingulate, but other areas, including the reward pathways, deeper brain structures, orbital frontal cortex, have each shown promise in the context of mood disorder and major depression. So other things that we keep our eye out when we think about this is how depression is thought of as a disease. And previously, a lot of the categorization of depression was based off of the symptoms, the outward manifestation of this disease. However, this doesn't necessarily track with the neurophysiology underlying the condition. So there was a very nice broad meta-analysis done by Leanne Williams, largely focusing on prior studies in imaging. And she was moving away from the symptom-based categorization and instead was looking at specific network dysfunction underlying aspects of depression. And she came up with these depression biotypes, or different networks that may be involved in the dysfunction. And then started looking at if different therapies might best target these different networks, and then which symptoms related to these networks. So we can see, there's a number of different large-scale intrinsic and task-evoked circuits from prior published work. A number of these are going to be familiar to us: the default mode network, salience network, both positive and negative affect networks. And by looking through the literature, she found that a number of these networks seem to be implicated in core symptoms of depression. Specifically, rumination seemed to result from dysfunction of the default mode network. And you can see with these other key core deficits and problems in depression. And many of them involved multiple neural networks. So at this point, taking all of this together, we designed the Presidio clinical trial. So again, this was a personalized closed-loop deep brain stimulation trial for the treatment of depression. There's a few ways in which this differs from the current field of how DBS was being applied in MDD. So first, we use responsive or closed-loop stimulation applied during periods of high symptoms. And we identify those periods of high symptoms by a correlative neural biomarker. We have a 3-stage design where we find personalized stimulation targets and the best neural biomarker of symptoms in order to program that closed-loop stimulation. And we're using, in stages 2 and 3, the chronically implanted NeuroPace responsive neurostimulation system. So this is a bidirectional, cranially implanted device. It's been FDA cleared for the use in epilepsy, but we have an IDE to use it in the case of MDD. And you can see here in this illustration, the can is cranially located under the skin, and there are 2 4-contact electrodes that are implanted. We use depth electrodes for both of these, with our goal being one is delivering stimulation and the other one is sensing a programmed pattern of neural activity that serves as our closed-loop control signal. So very briefly, going through the 3 stages of this trial. In stage 1, this is a relatively short stage in terms of duration, but it's a very data-rich period. So here, patients are admitted to the hospital. They're implanted with intracranial EEG electrodes, 10 electrodes, 5 on each hemisphere. And in the context of being inpatients in the epilepsy monitoring unit, we do continuous recordings for 10 days. We'll work with them for 12 to 16 hours a day, testing different stimulation parameters, recording activity to try to correlate with their symptoms to identify a biomarker, and doing a bunch of other different tasks to assess their baseline features of depression and then how stimulation may be moving the needle on those. Continuation into stage 2 is contingent upon us having identified a personalized stimulation site that seems to alleviate symptoms. And then in stage 2, they are implanted with the RNS system, at which point we can have them record short snippets of neural activity in their home environment or when they're ambulating. They no longer have to be in the hospital with us. So the core goals of stage 2 are to identify a neural biomarker of MDD symptom severity, and then to test that biomarker in the context of the closed-loop algorithms that the RNS system is capable of implementing in order to deliver the most beneficial individualized stimulation program that we've identified. And then in stage 3, which begins either after 1 year has elapsed in stage 2, or after we find a closed-loop regime, which reduces the MADRS score by at least 50%, maintained over 4 weeks. The MADRS is one of the standard outcome measures in depression. They'll enter into a double-blinded crossover period, which I'll elaborate on more in a bit. And then following stage 3, they're in a long-term follow-up period where they're allowed to keep the device. They can stay in the programmed manner, or they can be explanted per the patient's desire. So I'd like to introduce you briefly to the 3 patients whose data I'll be discussing today. PRO1 is a woman in her late 30s with severe treatment-resistant MDD that had a childhood onset. She has a family history of suicide, 3 distinct lifetime episodes of depression with better functioning interleaved. And her most recent depression episode lasted about 4 years, and it included mainly symptoms of anhedonia, energy, and cognitive deficits, which at times did require hospitalization. This specific episode was not adequately responsive to 4 antidepressant medications, augmentation strategies, ECT, or TMS. So because of this, she qualified for our trial. PRO3 is a transgender male in his early 30s. He was first diagnosed with MDD at age of 12. He also has a family history, in this case, of severe depression. He's college educated, started medical school, but had to leave because of the severity of his symptoms. And he has also had extensive prior treatment history, trying multiple different categories of medications, including esketamine, and then also undergoing ECT in the context of being hospitalized. And one feature of his depression is recurrent suicidal ideation. And then the last person included in today, whose data is included today, is PRO4. Now, this is a woman in her late 20s with symptoms onset in her mid-teen years. She's had a challenging home environment for her whole life, and she was previously a very accomplished athlete who also had started a graduate program, but had to leave because of the severity of her symptoms. She's also tried 4 classes of medications at maximum dose, extensive psychotherapy, both IV and oral ketamine, multiple sessions of TMS and ECT. And she's had multiple prior hospitalizations, largely because of suicidal ideation, and multiple suicide attempts. So as you can see from these very brief medical histories, we're talking about people who have had MDD for quite a large portion of their lives and who are not being helped by current therapies. So again, very briefly, stage 1 involves this 10-day hospitalization. This is just a screenshot of our typical schedule. So we really try to do as much as we can when we have those intracranial EEG electrodes implanted. So the big distinguishing factor between this stage and the future stages is here we're using 16-channel PMT electrodes. So we have 160 contacts across both hemispheres of the brain in all the regions listed, and we can do 24/7 neural recordings at this time. So this gives us the largest spatial coverage that we're gonna have of multiple different brain regions at any point in this trial. And we use this stage to identify which brain regions are gonna be most effective for our chronic implant, which again, as a reminder, is only 2 leads with 4 contacts each. So we do expansive stimulation testing during this time period. And just as an example for the types of results that we hope to have during this time period, this is a graph of PRO3. And here on the y-axis, higher values indicate worse symptoms. So this is a visual analog scale of anxiety, a visual analog scale of energy reversed, where high energy is good, but we've reversed it to plot it such that high symptoms are bad. Those are all higher values on the plot. This is a visual analog scale of depression. The HAM-D6, a condensed version of another standardized depression outcome scale. And what I'm showing is different time points at which the patient responded to this suite of surveys. It takes about 1.5 minutes for them to do on an iPad, so we can have them do it multiple times throughout our testing. And excuse me. And in this case, we were doing what we call intermittent stimulation testing. So again, one of the things that differentiates this trial from previous therapies is we are not applying stimulation continuously, but rather when we get to stages 2 and 3, we'll be applying short periods of stimulation in response to a biomarker. So in order to mimic that, when we're trying to identify the most beneficial stimulation sites and parameters, we do this intermittent stimulation testing, where we artificially have stim on for a certain duration and off for a certain duration. At this point, we don't yet have a biomarker identified, so we can't use that to help us in our testing. But over the course of this testing, when we were changing the parameters of the stimulation, we can see that what started as a relatively moderate to high symptom state decreased, and then this black line indicates when washout started. So we did not tell the patient that anything changed, but we stopped administering stimulation. The goal of this to be to see how long the sustained effects of stimulation were. And in this case, we were able to maintain effects for up to a couple of hours. And you can just read some of the verbal reports that the patient spontaneously gave to indicate how this was for him. This was similar testing at a different site, and we can see first we were changing parameters and we were not very good at finding a therapeutic stimulation site, but at some point we had a precipitous decrease in symptoms. And this is, again, useful for us to understand the amplitude of stimulation or the pulse width of stimulation that's most beneficial. But then following a washout period, those symptoms rebounded, at least to some extent. So when the stimulation was working, it was working well, but it doesn't work all the time. And one of the things I'd like to highlight here is we're not trying to artificially make people happy, but rather this verbal report of, I don't feel as overtly happy, but I feel a lot more functional and happy to exist. I feel like how normal people live. This is kind of sensation I've been missing for years and years. So we're really trying to remove the block that these people often have in order to function and achieve their activities of daily living or be able to hold a job. The goal is not to make them feel good all of the time, because of course that's an artificial, non-physiological state. And then just one example from our other patient, just to show this is not an N of 1 type phenomenon. So this is PRO4. And here we were doing stimulation in the left nucleus accumbens. Just because of how the trajectory for our left subgenual cingulate contact worked, we were able to target this nucleus accumbens region as an added bonus. And here, similarly, high values are high symptoms, low values is what we're trying to achieve. We tried of the same contacts, a few different on-off duty cycles of the stimulation interleaved with sham or periods of no stimulation. And we can see that during periods of sham stimulation, these first two symptoms were pretty high, and they decreased during various rounds of testing. And then there was a slight increase at the end. This patient did not spontaneously provide verbal reports about how she was feeling, but you can see we have annotated the behavior that we observed from her. So moving on to stage 2, this is where the NeuroPace RNS system is chronically implanted. And following that implant, we have each of the patients do 2 daily at-home patient neural recordings concurrently with symptom report surveys. So this allows us to build up a data set where we can then look and see what neural activity is correlated with periods of high symptoms or low symptoms. This is also a period where there's frequent study visits to test and optimize stimulation parameters and to update the biomarker detector settings. So these are some results from PRO1 from her stage 2 period. Again, one lead was used for sensing, the other lead was used for stimulation. And as we collected data, we were able to find that specifically gamma power in one of her amygdala bipolar recording channels was positively correlated with the outcome measure of HAM-D. So this allowed us to use gamma power as the control signal to program our detector to then trigger what ended up being VCVS stimulation to be the most therapeutic stimulation. So we can see here the change in the visual analog scale of depression. There was the largest decrease with the VCVS stimulation. And again, the system that we're using does not provide continuous stimulation, but rather we program it to have a fixed duration of stimulation each time the detector is triggered. And we found that a burst duration of 6 seconds of stimulation was sufficient to decrease symptom severity. And then lastly, stage 3 is a double-blind crossover period where we actually have a double-blind randomized crossover at the beginning of the stage 3 year and at the end of the stage 3 year. During this time, we have the patients on 6 weeks of their optimized closed-loop therapy and 6 weeks of sham, conditions counterbalanced and blinded to them. And our primary outcome measure is the change in MADRS score across those 2 6-week periods at the beginning of the double-blind randomized crossover. So this is the backdrop of the Presidio study. And I'm going to be using data from this to now go through 3 vignettes talking about some of the key questions that we have in doing this type of work, how far we've gotten in answering those, and then just posing some questions and directions that we need to keep working, and how additional collaboration and tools are going to be necessary to keep making that progress. So the first vignette is, where are we? And here we're saying, what is stimulation targeting? So this work was spearheaded by Leah Sugru, who is a neuroradiologist who works with this group. And Leo applied methods from Andreas Horn and Andrea Kuhn, specifically implemented in the LEAD DBS toolbox. And here he was trying to model the volume of tissue activated, or the VTA, and conduct tractography. So the volume of tissue activated can also act as a seed to model the cortical structural connections from the region that's being stimulated, as well as the cortical functional connections. The data that I'll be showing today, the structural connectivity, is based off of a normative connectome informed by the Human Connectome Project, including 30 to 40 healthy participants. And the functional connectivity is based off of resting state data from 15 normative participants. So starting with PRO1, we could take a few different regions that showed very promising effects of stimulation and use those as the seed regions for the volume of tissue activated. And then by that volume intersects with white matter tracks, and we can then see where those white matter tracks, first what they are, and then where they project to. So in this case, when right ventral capsule stimulation was applied, we see that we hit white matter tracks going to the amygdala, some pathways to the fornix. When we use the left genital cingulate stimulation, we see that areas such as the stria terminalis and the cingulum bundle are targeted. I would like to mention that the therapeutic stimulation that we applied involved bipolar stimulation between either adjacent or non-adjacent contacts. However, the way that the software is currently designed, it's very difficult to do model bipolar stimulation, which is what we actually experimentally applied. So rather, there was just a sphere of activation modeled around a given contact acting as the anode, and then the can was serving as the cathode, or the positive contact. And then using these regions of volume of tissue activated, we could then use the white matter pathways to project and look at both the structural connectivity and the functional connectivity at the cortical level that was implicated from this subcortical stimulation. Again, this was using data informed from the Human Connectome Project. And on the left, we're seeing these are the regions where the tracks are projecting to. And then on the right is the fMRI resting state functional connectivity. And for those familiar, when you look at these activation maps, the first thing that you may see is a striking similarity to the default mode network. So the default mode network is connectivity between regions that has been identified in task-free conditions where individuals are asked to rest and reflect. And the default mode network, or alterations to it, are actually highly implicated in major depression. Prior studies have shown that there is hyperconnectivity of the default mode network with a number of other regions, including the medial prefrontal cortex, the anterior cingulate cortex, that are involved in emotional thinking. And that's thought to potentially be one of the mechanistic reasons leading to rumination in a lot of these individuals. And in addition to this alteration in strength of connectivity, there's also been shown to be higher variability in the connectivity. So the stability of the default mode network may be fundamentally altered in individuals who have MDD. Furthermore, other treatments seem to modify how the default mode network works. So TMS has been shown to increase the strength within certain regions of the default mode network, particularly the role of the dorsolateral prefrontal cortex, thereby kind of normalizing that inter-network connectivity and functioning. And then ketamine has been shown to disrupt the elevated connectivity between the default mode network and some of those other networks implicated in emotional regulation, thereby potentially decreasing some of the ruminative thought. Very briefly, just to show we could do this with other patients. So for PRO3, again, showing which white matter tracks are implicated for 2 different stimulation sites. And again, we see those same default mode network regions implicated here. So what are some improvements needed to where we are right now? So first, as I mentioned, we're relying on the anatomy from normative controls. So we're currently in the process of applying these analyses to individual patient space. So a one-to-one match with where the stimulation is taking place in the individual's brain and their anatomy for what their connectivity looks like. But perhaps more fundamentally, this VTA is a very crude approximation for what actually is going to be happening when we apply electrical stim. Unfortunately, it's devoid of biophysically realistic current propagation. It just assumes current is spreading radially from the source, which we know is not going to be the case because of the orientation of electric fields relative to cortical layers and the different cytoarchitecture of the cells involved. Furthermore, as I said, we typically apply bipolar stimulation, but this is modeling monopolar stimulation using an individual site and the can as the anode and cathode respectively. So it would really be beneficial to simulate true bipolar stimulation. And then that would also allow us to understand how reversing the polarity of stimulation might affect clinical results. Because we do see that flipping the anode and the cathode does produce different behavioral responses and different sensations, but we don't have a good understanding of why that might be the case in terms of the current flow. So next, vignette number 2, this is going to be talking about how's it going. So here, what we're talking about is how are we tracking the efficacy of treatment. There's a number of different ways in which we're currently doing this, and we're going to be focusing on one of those. So we have symptom reports. These can both be patient reports, self-reports for how they think they're doing, or reports from their consented caregivers. One of the interesting features of depression is patients will have clinic visits. They'll come in, their clinician will ask, how are you doing? The patient will say, awful, everything is awful. And their family member or caregiver will say, no, this person is doing fundamentally better. They left the house, they showered, they were interested in eating. So there seems to be a mismatch sometimes between what a patient reports and their observed behaviors. So we assess that from multiple sources. We can also track efficacy in terms of the neural biomarker. How well is this biomarker tracking symptoms? Is this biomarker returning more to a normative level? And lastly, we can look at network biomarkers. And by network biomarkers, one of the things that I mean is looking at cortico-evoked potentials, or CCEPs. This is work spearheaded by Ankit Kambudi, who is a postdoc with the group. So here, we're using electricity to elicit an evoked potential, similarly to how you might use a visual or an auditory stimulus to elicit an evoked potential. And these evoked potentials are very similar. They still have N1 and N2 components. One of the main differences is that we just need to remove this initial time period immediately following application of the electrical stimulus, because there can be an electrical artifact during that time period. But following approximately 10 milliseconds, we then have physiological activity. Here we apply a brief train of electrical pulses, typically at about 1 hertz, such that we can resolve the evoked potential following each of the pulses. And we apply this on one of our bipolar stimulating contacts, and then we record from all of the others. So here in blue, you can see this was the contact on which we were applying the electrical stimulation. It won't be shown for any of the subsequent plots because that's not physiological activity. But in the orange, red, and green traces, we have those physiological evoked responses. So we can do this network mapping over multiple sessions over the course of the study period in which an individual is receiving treatment or going through a double-blind crossover period. Highlighted here, we've just pulled out some of the trial averaged evoked responses across different mapping sessions, kind of in the context of PRO1's course throughout the study. And even just to the naked eye, you can look and see that the morphology of these evoked potentials changes and shifts over time. What were previously biphasic responses become monophasic, the complexity seems to decrease, and it's those morphological changes which are representing this network map that we can use. And again, we call it a network map as compared to a local response, because remember, we're stimulating in one region and then recording in all the other regions we have access to. So of course, there's the key question, are changes in this network structure related to symptom state? So across this time course, we also have those daily or multi-time a day symptom reports. And those are just color-coded here from 0 to 1 with lowest symptoms to highest symptoms. In general, we can see as the study progressed for this individual, depression in general, which is the second line, decreased over time with a few rebounds when we had double-blind crossover. But one of the questions that we'll be getting to in the next couple of slides is, are the morphology of these EPs somehow related to this symptom state? So how are we characterizing these waveforms? So I'll be showing two different ways in which we can do this. First is we can extract waveform features. So for example, we can calculate the root mean squared, the zero crossing, or the line length of this evoked potential waveform. And then very simply, we can look at correlations between those features and the symptom reports, in this case, that visual analog scale of depression. And for example, when we stimulate in one contact of the amygdala and record in another contact of the amygdala, we can see that we do have this correlative relationship between those EP features and our symptom reports. So if we expand the similar analysis to the whole network that we have access to, what emerges? So here we see that this specific one was calculated using the root mean square metric. And overall, we see that there's a reduction in an input-mediated response at amygdala 1-2 that was associated with decreasing depression symptoms. So by that, it appears that the amygdala was less excitable, for lack of a better way of describing it, as depression decreased. And then similarly, there was a growth in the input-mediated response in VCVS associated with this reduction. This is all well and good, but as you saw from the previous slide, we're picking out individual features of the morphology of these waveforms, thereby likely losing information over the course of this. So is there something that we can do that takes the whole waveform, therefore hopefully capturing more information to tell us a bit more about what's going on here? So Ankit employed UMAP, which is a uniform manifold approximation and projection. This was an approach developed which was actually applied to spiking data and the morphology of those spikes, and he converted it to be looking at EPs. And this really combines nonlinear dimensionality reduction with graph clustering. And it uses the EP waveform in its entirety. So it includes the amplitude, response timing, number of lobes, and then just overall complexity of this. So the goal was to somehow use this to inform symptom state, such that we can use that as a different type of biomarker. So here I've only shown a subset of the processing steps, But overall, this UMAP involves normalizing the peak-to-peak amplitude of each of these individual evoked potentials, calculating a nearest neighbor graph, which will project to a high-dimensional space, then creating a connectivity graph between those nodes, where each node is an individual evoked potential based off of proximity within the high-dimensional space. And then using graph clustering community detection to identify the nodes that are highly similar to each other, but then dissimilar to nodes that are grouped in other categories. And then lastly, we can visualize this as a high-dimensional graph, visualize the high-dimensional graph as a 2D manifold. And that's what's shown here, this 2D representation of the manifold. And we can see, it seems to do a pretty good job of automatically categorizing the different EP waveforms and then having the trajectory move from certain features to other features. So this is all well and good, but one of the things that's really important to us as we're programming stimulation in these patients, as we're trying to treat them, is understanding how some metric such as this actually applies back to their symptoms. And so we look to see how correlated the different clusters of this EP morphology were with the self-reported depression. And what we found was that there was indeed a relationship between those different clusters and the level of depression that the individuals were experiencing, where some of the more complex waveforms were associated with higher depression and the more traditional, responses to the EPs were associated with lower depression. So this makes us start to think that we can run this evoked potential paradigm and use that as a more objective readout or a non-patient self-report involved readout for the state of the network and how the patient is doing. So next steps for this type of treatment, what I just outlined is how this model can inform advancement in treatment. And then of course there's a number of different questions related to the results in and of themselves. How stable is that manifold? Ideally, we want to move from one category to another. So what perturbations facilitate that movement? How is the stimulation that we're applying related to, going through that manifold? Can we find a shortcut and not have to go through each of the different states, but rather, take a highly symptomatic person and get them into remission sooner? And then very briefly, I'm going to go through our last vignette. So this is it working now? And this is looking at variability of therapeutic effect. So one of the things we found with PRO1 was there was actually a state-dependent response to stimulation. And in this case, it was pretty clear that this state-dependent response was based off of arousal or energy level, where we found that the pre-stimulation arousal level really influenced whether stimulation was beneficial or detrimental to their symptom state. So for example, 1 Hertz stimulation in the OFC, if it was applied during a low arousal state, it actually worsened mood. Whereas if it was applied in a high arousal state, it improved mood, basically calming the person down from an overly anxious state. Conversely, VCVS stimulation improved mood independent of the starting state. This was one of the reasons why VCVS 100 Hertz stimulation was the stimulation that we advanced this person on into stage 3. However, what do we do if those different states aren't as self-evident and we can't actually figure out what they are? And this is something that we are currently trying to navigate with some of our other patients. So this is just a visit summary from one of our recent study visits with PRO3, where you can see we tested a number of different stimulation sites and parameters. And over the course of those applied stimulations, we basically pulled everything together and said, did the patient improve or worsen over the course of that stimulation? We applied each of these for multiple days for replicability, and we can see that the story is really mixed. There was only one site which seemed to work on both of the days that it was tested. But with this person, we have not yet been able to identify what the state dependence might be of this. Furthermore, this patient also experiences very profound fluctuations in their symptoms during periods of no stimulation or sham stimulation, where again, they have low symptom states, followed by higher symptom states. And you can also just see a lot of up and down, a lot of noise in this graph. And this is reflected in their self-report, in their diary entries, in their verbal reports. So this is very real to what the patient is experiencing, but it's complicated in the context of how do we know what the underlying variability of symptoms may be if we then want to understand what a therapeutic effect might be of our stimulation. So overall, it's infeasible to exhaustively test stimulation in all of these different states. So we're thinking about and working on developing whole brain models of these different states, and then hopefully trying to relate the state, the neural properties, and the symptoms. And then in that model, apply current to simulate the stimulation and thereby understand how we may be shifting from one state to another or changing the properties of that state. And overall, we think it's very important that this be informed by empirical data. So we can look and see, well, what changes are induced in neural activity by the stimulation, and use that to validate the model. I think use our collected data to make sure that the model overall is ecologically valid and actually helping us inform future treatments. So in summary, we went through where are we going, the tractography for trying to understand where in the network our stimulation is reaching. How's it going? Looking at tracking changes in the network, looking at that network biomarker as created by the evoked potentials, and then very briefly talked about, is it working now? How to navigate variability in response. So looking ahead, we just think that, or I think that tools beget need, which beget tools. So we had these new tools for being able to apply this closed loop stimulation in people to get this ambulatory data. Now we have a need for how to analyze those data, and then that's just going to beget tools to be able to answer those questions. And all of this is working towards informing circuit-targeted precision medicine. Thank you very much, and we might have time for a few questions.
Thank you for this fascinating talk. So I'm very curious, what do you think is the mechanism of action of this deep brain stimulation? So is it that the stimulation is doing some kind of virtual lesion or reducing the activity of the target area? Or what is the kind of mechanism?
Yeah, the mechanism of electrical stim is something that I really wish I could stand up here and say, it's this definitively. I think it's a question that strangely is not well understood at this point. So one of the things that we found is the frequency at which stimulation is applied very much seems to change how the network is responding in terms of the neural activity as well as the symptoms of what we're seeing. And that makes sense from a lot of the very early work done in long-term potentiation and long-term depression. So I think dependent upon the frequency of stimulation, you may be doing a very brief lesion type effect of the region. You may also be increasing the excitability of a region, leading to kind of the equivalent of hyperexcitability, where incoming input will have more influence. Overall, what we see is the effects of stimulation can outlast the stimulation, but at this point, it does not appear to be permanent.
Thank you.
Thank you. Fascinating talk. Just wondering, you showed us the brain state when you use the perturbation. I'm wondering for different patients, do you see the similar trajectory between the states, or there's vary between patient to patient?
So are you referring to like the UMAP type work of movement through those states?
That's right.
Right, yeah. So at this point, we've only really seen kind of long-term unidirectional movement through that space. That being said, what I showed today were results from our first patient, where we had relatively sparse sampling of our overall experimental time course. But given that it seems to be promising, we have drastically increased the frequency with which we're doing that experiment, such that we can try to have more longitudinal time points through there. So stay tuned. What we're hoping is for individual different stimulation programs, when we test them, some will work, some won't, and we'll see movement through that manifold, which can help inform us, is this stimulation paradigm working? So we've increased how we're doing those experiments to hopefully be able to get towards a better answer for that.
Thank you.
Hey, thank you for the talk. It was so interesting. I was just wondering, during the trials, were the patients receiving other kinds of treatment?
Yes, so patients are receiving other treatments for their depression. We ask them, and we work with their psychiatric care teams to try to maintain stable treatment throughout the course of the trial. So that includes both maintaining the same pharmacological treatment as well as any of the same therapy or psychotherapy interventions that they may be receiving. With the types of patients that we have, suicide is always a high risk, so safety of the patients is always front and foremost. So therefore, we don't consider it ethical to take them off of any antidepressant treatment that they may be on aside from the stimulation. So that does add some variability across our patient pool in terms of, are there effects and interactions between the different treatments. And I think that's just something that time will have to tell as we increase the number of individuals that we're working with.
Cool. Just one more question. With those biomarkers, are you looking at other ones that aren't related to the overt potential?
Yeah, so there's a large body of work that we're doing looking at other biomarkers. So first we look at spectral power, and those are actually the biomarkers that we're then programming into the device to control stimulation. We also have our patients wear Fitbits, so we're collecting peripheral physiology type data, heart rate variability, step count, parameters about their sleep, and we're looking to see how that may correlate with symptoms, and potentially combine the self-report, the neural activity, and this peripheral physiology to try to give us a more cohesive picture of what's going on.
Cool. Can't wait to see more results. Thank you.
Stay tuned.
Thank you for your talk. I have a question about frequency of stimulation. You mentioned that different frequency, different response. And I guess follow on that, did you try to use patient-specific frequency, for example, based on the patient's peak alpha or something like that?
At this point, we have not done looking at the patient's resting state spectra and choosing specific frequencies from there. Very interesting idea. I know that it's been, very advantageous in other areas, so that's something we could work on. One of the things that we've recently started to do is look at some of the biomarker activity in the spectral domain, and let's say gamma power is most highly correlated with symptoms, trying to think, given the gamma theta relationship, could we stimulate at theta to try to decrease or suppress the gamma, thereby moving from, if you have a positive correlation, high gamma, high symptom, lower gamma, lower symptom. So trying to take advantage of some of the brain's natural relationships. One of the challenges that we face is different frequencies of stimulation have higher likelihoods of inducing seizures. So none of the patients that we work with have epilepsy, but when you put electrical current into the brain, you're always at risk of inducing a seizure. So we need to be mindful. For example, 50 hertz stimulation, for whatever reason, seems to be a bit more seizure-genic. So we just need to be mindful of like safety boundaries.
So you go above 24 hertz that's usually used?
Sorry?
You're going above 24 hertz stimulation?
Yeah, a lot of our stimulation is at around 100 hertz, give or take.
I see.
But we have a pretty broad range in which we can apply it.
Thank you very much.
Thanks for the great talk. This is maybe gets a bit outside of medicine, but I guess with depression, there are more than just cognitive symptoms. There are sometimes like more overt behavioral symptoms. Do you know if there are animal models for depression that to begin with, and then maybe you could try different stimulus protocols on those models?
Yeah, this is actually a very important thing. So a lot of the work that we've been doing is funded by a Dolby Family Center grant. And that grant funds like a set of human work, but then also a set of animal work that's done both at UCSF and at Stanford. So there's a number of groups there that have genetic mouse lines mimicking different symptoms of depression, and they are very much in the process of looking at detailed circuit pathways of depression and then how perturbations such as this might influence it. I don't have the specifics right now of what they're working on, but that line of investigation is definitely taking place.
Oh, okay.
Just wonder what you think of the role in, some sort of patient selection based on pre-management imaging, like you showed electrodes in the BTA. Helen Mayberg tried the subgenial ACC. So depression is a very heterogeneous disorder, as you know, I'm sure. And in addition to the phenotype, there might be an underlying neurobiological differences. And for example, we saw in some of our studies, some patients lose a lot of activity and variance in different brain regions like the subgenial ACC, and others don't. And I wonder what you think about preoperative or pre-interventional sort of imaging with mood induction in the scanner and patient selected and that whole repertoire of choices.
Yeah, I think this is actually very important, and it's something that we're collecting data on to try to inform a bit more information. So each of our patients will undergo 3 fMRI, DTI, and just MRI scans prior to their first implant. And we're basically doing that to try to collect data such that after we have our full cohort, we can look back and see, were there consistent structural networks or consistent functional networks, which then correlated with success of the DBS treatment or not. We've also recently been talking to a prospective postdoc who does whole brain modeling, and she's been very excited about the prospect of using those data to then try to understand which networks might indicate greatest efficacy of the treatment in future. In general, this is a very invasive approach, right? It requires multiple neurosurgeries, very time-intensive. So any sort of non-invasive equivalence that we can come up with or non-invasive screening that we can do is definitely going to help us make sure that this treatment reaches the right population.
So is there any experience how long this impact lasts, the positive impact, when you stop the treatment? So that's an important issue, I guess.
Yeah, so that's still not fully elucidated at this point. So in the stage 1 testing, the patients are inpatient, we have seen up to 2 to 3 hours of beneficial effect after we finished stimulation. So on the one hand, that's great. From a closed loop perspective, if you apply a perturbation, then you have a couple hours where you don't have to be applying stimulation. That sounds good. In terms of what information we have at this point in terms of true longitudinal data, Piera-1 is the furthest along in the study, and she has had control of her symptoms for over a year, but that's with the adaptive stimulation enabled. She receives approximately 30 minutes of stimulation interspersed throughout waking hours, and I am not at liberty to divulge the double-blind crossover results, given that we still have study staff who are blinded, but we do have those data for what happened when we did have her on sham periods. So stay tuned for when we're able to publish those. Thank you very much.
Alrighty. Hi guys, my name is Daniel Petrovsky. I am a PhD student at Swinburne here in Melbourne under Tanya. Don't be afraid to tell her how good I'm doing. Still early in my PhD, so make sure she keeps me on. Couple announcements. There is morning tea right after this. I'm also the student coordinator on the local organising committee, so in charge of all the volunteers. I'd like to take this opportunity to thank all the volunteers for doing such a good job. Some of them are here. Yeah. Tonight there is an after party at State of Grace. On the back of your lanyards you'll have 2 drink vouchers. It's also got the address there. It's from 7:30 on till late, so feel free to come anytime. If you need directions on how to get there, there's always Google, but you can always come to the information desk out the front. We've got some little maps there that we can hand you over to help you get there as well. Okay? Enjoy the rest of the day and we'll see you outside.